Hello, are you seeing something?
Yes, it's
Yes, it's
visible.
visible.
Oh, it's visible. Thanks very much. You can see the topic weed and and crop interactions in agricultural systems.
Yes, we can
Yes, we
see,
can
Joe.
see, Joe.
Thank you very much. So, in this lecture, we want to understand how weeds can actually affect the crops. So the aim is first to understand how weeds affect crop growth and subsequently reduce crop yields. This information could be useful for planning weed control programs in order to maximize crop yields. So the question we are addressing is, how do weeds cause crop yield losses? You may be aware that crops and weeds share the same environment where they obtain water, nutrients, light, oxygen, and the carbon dioxide for their growth and survival. So sometimes these resources, specifically water, nutrients, light, maybe oxygen and carbon dioxide, may become limiting, and as a result, probably the crops may suffer. So weeds generally adversely affect crop growth, leading to reduced crop yields. Of course, there are some crop genotypes which can also suppress the growth of weeds. Here we have got a slide showing us a maize crop with the weeds. This is specifically, we have got blackjack, which is infested, in the maize field. Here you can hardly see the soybean crop in a weedy environment. Of course, you can see those lines. I hope they are visible from where you are of the soybean, but there are also weeds. So, how do weeds interfere with crops? There are various processes. First, weeds can actually compete for the limited resources. So we have competition, or weeds may actually produce some biochemicals which can negatively affect the crop. Here we are now talking of number 2, which is adenopathy. Now, when we have two processes, that is competition and arenopathy occurring together, we have what is known as interference. So normally weeds interfere with the growth of crops. In fact, under natural conditions, we have these two processes operating simultaneously. And in reality, we do have interference, although sometimes we talk about competition and arenopathy separately from this interference. The other issue is where you have the wheat, which is directly benefiting from the crop, and there is actually a physical bond between the weed and the crop. Here we are talking about parasitism. So please remember, when we talk about the effects of weeds on crops, there are these four processes: competition, arenopathy, interference, and the parasitism. Now, we want to focus specifically on competition. In fact, we shall be talking about competition most of the time. But of course, later we'll talk about arenopathy and parasitism. So what is competition? This is the process whereby the crops and the weeds engage in a sort of a struggle to get limited resources, which are specifically light, carbon dioxide, oxygen, water, and nutrients from the environment, usually resulting in a reduced crop growth. So this is the concept of competition. What you should understand is that there are resources which are limited. Either the crop or the weed is trying to get hold of those resources. Well, this is the definition which was given by Grime. And of course, there is no need to memorize this quoted statement. It is the tendency of neighboring plants to utilize the same quantum of light, ion of mineral, nutrient, molecule of water, or volume of space. So probably this should also remind us that weeds and crops also compete for space, which was not apparent in the first definition. So here, just to remind you that we are talking about weeds and crops. Here we've got a groundnut crop with weeds. In fact, we can hardly see the groundnut crop. Maize crop with weeds. Probably a lot of things have happened here. You can see the maize crop is already tasseling, even before knee height. There are also nutrients which could be a problem, which makes actually competition very intense. This is a weeded groundnut crop. So what can happen when you have wheat is that you have the crop expressing some nutrient deficient symptoms. If you look at the color of those maize leaves, they are purplish, probably indicating the phosphorus deficiency. They could be also be yellowish, which could be nitrogen deficiency. So in this case, it could be a combination of two major nutrients which are deficient in the maize crop. Probably the weeds in the background are taking a lot of these nutrients. Here we've got again nutrients deficiency. Here we've got the maize exhibiting those characteristic colors, purplish and yellowish, probably indicating the absence of phosphorus and nitrogen, which could have been taken by the weeds. Again, purplish color. Here it's a characteristic of phosphorus. Here we've got the height of the weeds and maize more or less the same. Probably they are competing for light. Again, here probably there's competition for light. You can see some shading in the background where it is dark, which means light is being intercepted by these plants. Here, believe it or not, it's a commercial field of soybean, but what we can see are just weeds. We can hardly see the soybean. I won't mention where we saw this field. Again, you can see the soybean, which was seriously affected by weeds. Now, in some cases, we see couch grass in fields. Where you see couch grass, there's a characteristic impact on the maize. You can see here maize with very short stems, and along with it we can see the Cynodon dactylon. Here probably we can say that yes, there is competition, but there could be also arenopathy. This could be a typical example of interference, which is caused by Cynodon dactylon. Here's the field of weeds, of maize with weeds. This is in Murewa. Just to make you aware that weeds can be a serious problem under field conditions. Again, that field seriously infected with weeds. Here you can see somebody who is weeding. The field seems to have a lot of weeds. After weeding, there's very little of the crop. Again, another field with weeds. Okay, the same thing here. Maize infested with grasses, which are weeds. Here we've got Acanthospermum hispidum. What I want to emphasize is that later on we may be trying to quantify what weeds can do to the crops. Sometimes we use quadrats where we count the number of weeds in a quadrat. That is that wooden piece of equipment which is on in the field. Sometimes you can take biomass. We will talk about this later on. Just to mention a bit on allelopathy, weeds or crops could produce harmful substances, which we call allelochemicals. These are produced via leaves or roots, which can adversely affect the crop, the growth of either weeds or crops. Well, when we talk of interference, this is, as I have indicated, it's the combination of competition and allelopathy. And this could be the real situation occurring in cropping systems under field conditions. However, weed competition studies frequently reported in literature could be correctly described as interference studies. There's also this term, we're not going to talk about this thing, commensalism. Is the relationship between unrelated organisms in which one gets the benefits from the other without harming it. But we are mostly interested in a relationship where crops are likely to be harmed by the weeds. Parasitism is the extreme case where a weed can exploit a crop. This situation is where a plant depends on another plant for its carbon and nutrient requirements. The growth of the host is adversely affected. Later on we will talk about parasitic weeds, but here I have got 5 examples. Number 1, Striga asiatica, which can be attached to a maize, sorghum, or pero millet. Then we have Electra vogeli, which can be attached to cowpeas. Striga forbesii, again, which can be attached to maize, sorghum, and millets. Striga gesneroides can specifically attack tobacco and cowpeas. Then we have got Cascuta species, which can attack to lucerne. So these are the weeds which can cause parasitism. Striga asiatica in sorghum. This is the Chundura communal area. You can see the red flowered plant in the background. Again, a closer look at Striga asiatica parasitizing maize. So let us move on to plant competition. We have got 2 types of competition. We have what we call above ground or aerial competition, which can be competition for light and carbon dioxide. Then we have got below ground or subterranean competition, which could be competition for water, nutrients, and oxygen. Then we've got forms of competition. We've got intra-specific competition. Same plant species can compete. For example, maize versus maize. It's like politicians in the same political party competing, you know. This is intra-specific competition. Then you have inter-specific competition where you have different plant species. Here we have got crop and wheat. We could be having maize and Leucaena indica. Usually this is the type of competition which we talk about, inter-specific competition. So, what do plants compete for? We have just mentioned some of them. They can compete for light, that is above ground competition. Now there are certain plant characteristics which are important for the competition for light. High growth rate resulting in tall plants. We expect such a plant to be highly competitive for light. If such a plant has a high leaf area index, it's likely to harvest a lot of light. Now, leaf angle and arrangement are also important. You can have horizontal versus angled leaves. If you have got horizontal leaves, there could be a lot of shading. In other words, the plant may fail to utilize a lot of light, but if it has angled leaves, light can penetrate through. That plant is able to be highly competitive in terms of light. So usually we talk of a canopy structure which can allow light to pass through, or a canopy structure which can create a lot of shade and prevent light capture. Just have a look at a plot with maize and chamfer grass. You can see the maize is as tall as the chamfer grass, and there's a lot of shading here. Probably both plants are highly competitive for light, and in fact we can say chamfer grass is highly competitive for light. You can see a lot of shading in the background, which means that if the maize plant has got leaves which are displayed in the shaded area, this can reduce its photosynthetic processes. Again, maize and chamfer grass, you can see on the left, on the right side, there's a lot of shade. So that was about light. What about another resource, carbon dioxide? Well, we may assume that there's plenty of carbon dioxide in the atmosphere, and probably it's not likely to cause competition. However, let us look at the two types of plants. We've got what we call the C4 plants, which are said to be more competitive than the C3 plants. The C4 plants use PEP carboxylase and rubisco to efficiently use carbon dioxide. You can see most of our weeds, like Amaranthus hybridus, Cynodon, Cyperus esculentus, possess the C4 pathway, or the PEP carboxylase and rubisco systems. This makes them efficient users of carbon dioxide. We know carbon dioxide is used to form carbohydrates, which provide energy for growth, and this can facilitate high growth rates, maybe indirectly. Then we have got C3 plants, which only utilize rubisco. These have got limitations in utilizing carbon dioxide for the photosynthetic process, which results in carbohydrates, which are required for growth. So soybean, cotton, wheat and groundnuts could probably have reduced the growth compared to the wheat. Here I am displaying a graph showing the carbon dioxide in the atmosphere. You can see that carbon dioxide is actually increasing with the number of years due to our farming activities and other activities which could possibly add a lot of carbon dioxide into the atmosphere. So the question is, as we increase carbon dioxide in the atmosphere, are we going to alter the competition of plants? Now the assumption is that there is plenty of carbon dioxide, and therefore there is no direct competition for carbon dioxide, but plants are divided into three groups depending on how they can fix carbon dioxide to produce carbohydrates. We know carbohydrates are relevant for growth, and it is the growth characteristics which may determine how a plant is competitive or not. So we've got C3, C4, and the crassulacean acid metabolism, the CAM plants. The C3 plants constitute some of the crops, e.g. cotton, like I have mentioned, soybean, wheat and groundnuts. C4, we have got Amaranthus, Cyperus, Cynodon, but we also have maize under that category. The CAM plants, we have got these weeds like Portulaca, Oleracea, and the Fetian, Chivandi Chimuka. I mean, it's the same weed, sorry. Portulaca oleracea is the botanical name, Fetian common name, and our local name is Chivandi Chimuka. This is a typical CAM plant, which is also efficient in utilizing carbon dioxide. Here, probably what I want to say is that one of the problems which is endured by the C3 plants is the existence of a process which is destructive, known as the photorespiration. And this can occur under very high temperatures in the presence of light. Normally under these conditions, photosynthesis can also be speeded up, leading to very high concentration of oxygen within the plant. Now, this is where carbon dioxide is also fixed by the Calvin cycle. So we are going to have more of oxygen and less of carbon dioxide. And what will happen is that instead of the plant utilizing carbon dioxide to process carbohydrates, it will use oxygen via the ribulose 1,5-bisphosphate, which normally fixes carbon dioxide. This can actually lead to the reduction of glucose. Once glucose is reduced, it means that the growth of the plant may also be reduced, and the plant may not become competitive. So it is the conditions which can change, or the environment, especially high temperatures, can make the C3 plants inefficient in producing carbohydrates, and then they become less competitive due to reduced growth rates. However, if you focus on the C4 plants, there's very little photorespiration occurring in C4 plants. So they are making sure that carbon dioxide is always being utilized to form carbohydrates, and the carbohydrates are available for plant growth, and these plants become highly competitive. I think that is the message which is there. This also same applies to the CAM plants, which take their carbon dioxide during the night, because they have their stomata closed during the day, so that they don't lose a lot of their carbon dioxide. So they are also efficient in making carbohydrates, which enhances their growth, and they also become competitive. If you are interested in the carbon dioxide issues, there's a paper with weed interference with the field-grown soybean decreases under elevated carbon dioxide. So if you read this paper, it may give us an insight on the issues of carbon dioxide. But you can see that this is under experimental conditions, not under field conditions. Here's another paper, again under artificial conditions, where you deliberately just increase carbon dioxide, and you see how this can impact on the competition. Probably this is just an academic exercise, if you may want to call it, but it will later on explain what is likely to happen to our crops in future, because carbon dioxide is increasing. due to our activities. Now we move on to competition for water, which is quite direct. Crops in weedy fields or plots show moisture stress earlier than those in weed-free plots. Probably you could have seen it under field conditions. Like this previous season, it was a severe drought. So competition for water was serious. Competition is severe during droughts and in low rainfall areas, like probably natural region 4 and 5. We expect serious competition for water. It can also depend on the root characteristics, shallow-rooted plants versus deep-rooted plants. If there are differences in the rooting systems for the two plants which are competing, sometimes they may not interfere with each other because they are exploiting different areas in the soil. Competition for nutrients. This is common. Weeds and crops compete for nitrogen, phosphorus, and potassium, which are the macronutrients. Nutrient deficient symptoms are frequent, especially in weedy plots like yellowing of leaves, indicating nitrogen deficiency, and purplish leaves indicating phosphorus deficiency. So here we are again showing you the purplish leaves and yellowish leaves, probably highlighting that phosphorus and nitrogen are deficient. So this is competition. You have on one hand you have got the wheat, on the other we have got the crop, and there are resources which have to be shared. Above ground we have got light and carbon dioxide, and below ground we have got nutrients, nitrogen, phosphorus, potassium, and water. So in this first part of the lecture, we have just explored the type of weed-crop interactions, which are basically for competition, arenopathy, although we didn't talk about arenopathy in detail. We just mentioned it. And we said there is also interference, which is a combination of arenopathy and the competition. And the fourth thing is parasitism. So these are the processes in which weeds can affect the crops. But the key issue which is critical is competition here, where the weed and the crop can compete for light and carbon dioxide above the ground, and also nutrients, NPK, and water below the ground. Here you can see I have left carbon dioxide. Does it mean that it is not competed for? I mean, okay, it's there. But the question is, do plants compete for carbon dioxide? But mostly we are talking of nutrients and water, and possibly and and of course light. Now that we understand what the crops and the weeds compete for, the next issue is how do we measure the impact of weeds on crops? So that will be the next lecture. So I will have to stop here. Hello?
Hello, Dr.
Oh,
Chiripo.
ndafunga kuti ndindega kai.
Chiripo dog.
Okay. So what we're talking about is the effects of weeds on the crops. I mean, we've tried to highlight the processes and also what is competed for, which causes the crops to suffer during this type of relationship where we have weeds and the crops growing in the same environment. The next issue we are going to do is to explore how we are going to measure the impact of weeds on crops. So that will be the next lecture. Now, actually. Any questions?
Ndatoda kubvunza kuti pachifani chimuka chamamushora ndochiya chinoita paruva here kana kuti is different. Chifani chimuka
It's a fleshy sort of weed which we find mostly in the low veld. I think some people eat this chifani chimuka. Ndakazoti weed rinorapa here. I don't know whether you have heard about it.
Nowanza andachifunga pane chinoziva chinoita paruva reshe chinoti kukanaya chinoita maleaves green kana kukankurudira chinobva chaoma.
Okay. I know what you are talking about.
Okay, thank you.
Which is dry. If you put moisture, it turns green. That's what you are talking about.
Yes. Yeah.
Okay. So let us now try to shift to the other areas. Okay, can you see the slides? Hello?
Yes.
Yes, we can see them.
Good. So now we want to focus on how to determine the effects of weeds on crops. So how do we determine the effects of weeds on crops? I mean, this is very important. Once we understand these effects on crops, maybe we can be able to utilize this information to plan weed control strategies. So how do we do it? First, we have to do it through conducting experiments. The other issue is that we need to establish the weed density which can justify weed control. In other words, if we see weeds in a field, does it mean we need to remove these weeds? We also need to understand the competitive abilities, or to determine the competitive abilities of weeds, so that when we control these weeds, we can just target the harmful weeds. We also need to determine or to know the competitive abilities of the crop, so that maybe in breeding we may come out with tolerant crops which are not affected by the weeds. So this is the importance of determining the effects of weeds on crops. The first issue which we are interested in is to understand the effect or the impact of weed density on crop yield. How many weeds per square meter are critical in causing yield losses? In order to address this issue, there are a number of experiments which can be done, and this depends really on your interest. So the research designs which have been used to study the effects of weeds on crops are as follows. Number one, we have got what we know as the additive designs. Now, the additive designs have been specifically used to determine what we know, what we want to call the weed control thresholds, or simply the density of weeds which can justify weed control. So if we see weeds in a field, it doesn't mean that we should remove them. Maybe they are not harming the crop. They could be below this weed control threshold, the density of weeds per square meter, which can interfere with crop yields. We also have what is known as the replacement series experiments or approach. In this case, you want to determine the competitive ability of the crop as well as the competitive ability of the wheat. However, at this stage, I should say that even the additive design, you can also determine the competitive ability of the wheat. But the replacement series probably will give you more information if you are interested in the competitive abilities. I mean, number 2 can tell us whether a crop is tolerant or not to the weeds, which is also very important. Then we have got the addition series or response surface experiments. That one tends to combine number 1 and 2 into one design. And sometimes it becomes a bit complicated, but it is efficient in giving us information on densities which matter, also on competitive abilities of both weeds and crops. The dial is, here you are just looking at individual plant species. Maybe this one is for an academic exercise. Nevertheless, it may become also important. The neighborhood experiments. Yeah, when you look at a field which is infested with weeds, there are some weeds which grow along the crop row and others which grow in between. The question is, which weeds are more competitive, those in the row and those in between the rows? Maybe we should just remove the weeds in the row and leave those which are between the crop rows. Maybe they are not competitive. We can only get the answer by using the neighborhood experiments. So here are the 5 ways to measure the effects of weeds depending on your interest. So let us move on to the additive experiments, and I should say that these are quite important experiments, the additive experiments. The objectives or research questions which we are interested in: number 1, what is the minimum weed density which justifies weed control? This is very important, you know. It has got a bearing on the inputs which we can use to control weeds. What is the most competitive weed? We can also address this question using this experiment. Crop yield loss. We also want to understand the type of yield losses which are caused by weeds. So just remember the additive experiments. Here is an example of a study where somebody was trying to determine the effect of Rapoco density on maize growth. You can see, using just pots, you plant your maize, you also plant your wheat at different densities. Then you measure the response of the crop due to differences in wheat densities. Here we have got Tithonia rotundifolia, or wild red sunflower, effects on maize. Again, a pot experiment. Again, the same experiment. So here is the method of how you can go about doing the additive experiment. So we are talking about the additive experiments. So we are on the method of the additive experiments. Just remember that. Here are examples of treatments. You can have width density, which is varied. For example, we can have 0, 2, 4, 6, 8, 10, 12 plants, and 12 plants per pot. While crop density is held constant, you can have one plant for each crop. Treatments are replicated. This is a requirement for experimental procedures where you can have an RCBD or a CRD. The data collected could be crop height, crop biomass, leaf area, and weed biomass. But normally, in most cases, we focus on crop biomass. Then we can use graphs or regression analysis to evaluate the effects of weeds on the crop. Normally we don't use tables. We tend to use graphs in order to understand the effects of weeds on the crop. But sometimes people use tables or figures, which sometimes becomes, cumbersome. Now there are relationships which are used. Normally we relate weed density to crop yield or crop yield loss. Sometimes people get linear relationships. That is number one. Quadratic response is very rare, but you can get also exponential curve, which has been commonly reported. Hyperbolic curve, I'll also explain it. It has also been commonly reported. Then the sigmoidal curve. It depends on the range of your treatments. When you do the final analysis or regression analysis, you may end up with linear if you have got few treatments, or it can turn up to be exponential curve, hyperbolic curve, or sigmoidal curve. But anyway, these are just examples of relationships which you are likely to get. So, effect of weed density on crop yield. The questions which we are interested are as follows. What is the weed density which justifies control of weeds in order to avoid economic yield loss? Now it becomes a management tool if we can find that that value. The other issues are for weed management. Should we monitor weed populations in cropping fields in order to plan weed control programs? It looks like yes, we should do this. Is weed density the best tool for predicting crop yield loss? Maybe there are other issues which we should explore. I will talk about them. Is the data collected from one weed species applicable to fields with mixed weeds? Now, with this type of investigation or research, you are dealing with one weed at a time. But when you go into the fields, there's a mixture of weeds, and sometimes it becomes problematic to apply such data to field conditions. But at least we have somewhere to start. Here I just want to use a graph or a diagram which can show the relationship of weed density and crop yield. What you see there, which is curved, as we increase the weed density, at first the changes are not very they are not very big, but as you progress increasing the weed density, there's a sharp decrease in crop yield, and it tails off as you are at the highest weed densities. It produces a sort of an S-shaped graph. Of course, it's slanting, but it looks like a tilted S. This is the sigmoid curve, and it was proposed by a scientist known as Zimdar. If you look at the same weed density on crop yield loss, you get this kind of shape. Again, it's a sigmoid curve. What it is indicating is that at the beginning, when you have few weeds, the loss is very small. Maybe at that stage, you may you may not weed and get away with it. But if you delay, the crop yield loss will steeply rise up to a certain level where you have serious yield losses. Now, this is an alternative. This is according to Zimdar. You get this sigmoid curve, crop yield versus weed density, crop yield loss versus density. You also get that sigmoid curve. But some scientists think that if you relate crop yield and weed density, you get that kind of a curve. As long as you have weeds, there is no delay in crop yield loss. You will have reduced yields. Whereas this graph seems to indicate that there are certain weed densities where crop yield is not seriously affected. But this one is showing us that there is a great loss with few weeds. Again, you have, as long as you have weeds, you have serious crop yield loss. Now, with this kind of a graph, you can identify two areas. Maximum yield loss, which is represented by A on the graph, and the slope, which is represented by I. Now, those two parameters can be allocated to a weed, and they can be called competitive indices. So among different weeds, if we do a lot of studies, you end up with various competitive indices. You can actually identify weeds which you can say these are very competitive compared to others. So before I go very far, so there are two approaches. If we look at the relationship of weed density versus crop yield loss. This first one was proposed by Zimdar, who said that a few weeds, may not affect the yield seriously. This is what the graph is saying. Or at first, when you have few weeds, your losses are not much. So you can actually escape weed control strategies, or you may not invest in weeding if the weed density is such, if it is very low. That's what is suggested by this graph. This one says as long as there are weeds, you should remove them, according to Cousins. This one is a rectangular hyperbola relationship. The Zimdar one is the sigmoid curve. So Cousins rectangular hyperbola, and on the rectangular hyperbola we can identify two areas or two indices, A and I. A representing the maximum yield loss, and I the rate of loss or the slope, and these can be attached to a weed. So for a weed, we can know what maximum loss it can do and the speed at which it can reduce the yield can also be known. These are the two important parameters. Now the graph on crop yield loss, this one, we can represent it using an equation. YL is the yield loss equals to I, which is the slope, the slope which we get from this graph, and the A again from that graph, and the DW on the graph is represented by the weed density. So we can say yield loss is equivalent to the product, or we can just say slope times width density divided by 1 plus, or in brackets, slope times density divided by maximum yield loss, which is A. And that will give us the yield loss from that equation. Here is a graph. This one was drawn by a student who was studying the effects of Tithonia rotundifolia density on maize yield loss. You can see he also produced a rectangular hyperbola as was proposed by Cousins, which looks like that one. So this was done as it was a research project from a student here. And he went on to calculate A. Tithonia rotundifolia caused the maximum yield loss in maize, which was 64% or 64.3, which is A. The slope was, or the rate of loss was 1.48, according to that graph. So we can allocate these values to Tithonia rotundifolia. A 64, I 1.5. If we do another study with a different width, we can also allocate it these figures, and we can compare the two widths to determine how competitive they are, or how harmful they can be to the maize. So if they are growing together, we know which width to deal with. This was Oxalis latifolia density in Brassica napus. You can see that kind of relationship. Of course, at one, around one plant of Oxalis latifolia, there's not much damage. Again, this is when you plot yield loss versus Oxalis latifolia density. You get that typical rectangular hyperbola. Again, this is probably Leucine indica effects in maize. You get that sort of shape. Now, when the Cousins proposed the first equation, he said YL yield loss is equal to IDW slope times width density over 1 plus, or in brackets, I times density divided by A, which is maximum yield loss. That one there was creating some problems. What it means is that we are assuming that weeds and crops are germinating at the same time. If the weeds and the crop are germinating at the same time, then that equation becomes useful. But if the weeds germinate ahead of the crop, then that equation may become problematic. So Cousins had to look into that to take care of weeds which germinate earlier than the crop by modifying this equation to this. So it's the same equation. What he simply altered was 1 in that equation. Otherwise, we have yield loss is equal to 1 slope times width density over, but instead of using 1, he put an E representing natural log to the power of C times T. C representing the rate at which the slope changed, or I changed, and the T was time. Once he had done that, then that equation became very useful to predict crop yield losses. It could take care of time of weed seedling emergence into effect. So he substituted E to the power of CT by 1, which was not very good. But if you put E to the power of CT, then you can predict the yield loss in a field much better. It's the same equation, he just removed the 1. Here I've got a table with various weeds and references. So you've got weeds and the crop. Now for each weed in a crop, we have got its A value at the top there, and I value also at the top. So each weed has got 2 numbers which determine the competitive ability of the weed species in various crops. So we can compare those numbers and say which weed is more competitive. And down there you can see they've used the Cousins equation to come up with this kind of data. So what it means in this country, we need, for all the weed species which we have under different conditions, if we know their competitive abilities in various crops, various regions, probably it can become easy for us to design appropriate weed control strategies. Maybe we will need to use computerized programs, so that for natural region 1, 2, or 3, we come up with weed control strategies. But unfortunately, in this country, we do not have data to attach numbers to various weeds under different conditions. A lot of work needs to be done in that direction. Now, some people suggested that it's quite difficult to measure weed density. Probably it may be better to measure leaf area. you measure leaf area of the weeds and the leaf area of the crop. Once you have done that, you develop what is known as the relative leaf area, which we will use as a substitute for weed density. This was proposed by Croft and Spitters in 1991. So relative leaf area, which is LW, was defined as leaf area index of the weed divided by the leaf area index of the crop plus leaf area index of the weed. So you would use that value LW to in your graphs. Here on this graph, which is similar to that of cousins, we have a relative leaf area of weeds on the x-axis, then crop yield loss on the y-axis. You get that curve. You still have the maximum yield loss, which is A, but the slope, instead of putting an I, they decided to call it a Q. According to them, their equation becomes YL, which is yield loss, is equal to Q, the slope, multiplied by relative leaf area, divided by 1 plus, or in brackets, Q, which is the slope, over A, maximum yield loss, minus 1, or in brackets, multiplied by relative leaf area of the weeds. So according to these scientists, Croft and Spitters, they thought this was more efficient. Instead of counting weeds, maybe you are better off measuring leaf area, but it depends which is more time consuming. So this is the equation, which is the same. Y, which is yield loss, is equal to Q slope multiplied by relative leaf area, or divided by 1 plus, or in brackets, Q over A minus 1 multiplied by relative leaf area. And these scientists thought that probably this was a better approach. But it depends, people may just choose the method which is easy for them. Another method which is more or less similar to Croft and Spitters, you use what is known as relative cover. This one you can just estimate whilst you are walking in the field. You can estimate area covered by the weed, then area covered by the crop. Then relative cover becomes area covered by the weed over area covered by the weed plus the crop. Now we have a DPhil student who is doing similar work in Murewa, I think, and Mutoko, under 3 agroecological zones. The student is trying to use relative cover in order to estimate yield losses. She uses an instrument known as the canopia, where you simply take a photograph of ground cover, then from the photograph you get the percentage of relative cover, and you can measure your crop yields. And when you draw a graph, you can come up with this kind of thing: relative cover of weeds as the x-axis, crop yield loss on the y-axis. And the equation is yield loss YL is equal to Q times RC, relative cover, divided by 1, or in brackets Q over A minus 1 multiplied by RC. So the DPhil student, in her project, she's going to use this equation. It is going to be applied for maize yield loss and groundnut yield loss, covering natural region 2, 3, and 4 in Murewa and Mutoko. So we want to see the situation under a groundnut crop or a maize crop. Probably this information could be used by Agritechs to advise farmers on how to go about managing weeds. So this is the equation. Yield loss is equal to Q slope multiplied by relative cover divided by 1 plus Q over A minus 1, or in brackets multiplied by relative cover. When I did practicals with some students, they found that this last one of using relative cover was very fast, accurate, and less cumbersome, rather than counting weeds in quadrants and measuring leaf areas and so forth. So you may want to read this paper, A Simple Model Relating Yield Loss to Weed Density by Roger Cousins. If you can't get the paper, I may have to send it to you. Another one, Simple Model of Crop Loss by Weed Competition from Early Observation on Relative Leaf Area of the weeds. This one now by Croft and Spitters. The only difference is that Croft and Spitters are now using relative leaf area instead of weed density. Predicting yield losses caused by multiple species with composition. You may also want to read this, where we have various weeds under field conditions causing yield losses. This is much more realistic, maybe. Yeah, this is another paper. Right. Here you could have these references by Chivinge and Rukuni. who did a study here at UZ. Then the other one, Ali et al., 2013. Again on yield loss prediction models based on early estimation pressure. Of course, it is important to know how weeds can actually cause yield losses so that we can manage these weeds. And we said that we can use additive experiments in order to go about determining these yield losses. Now, these are the disadvantages of additive designs. Crop biomass varies with the species proportion and density. The competitiveness of the crop is not known. It cannot be determined. That is the problem. This one cannot separate intra-competition among weed species or maize species or crop species and interspecific competition. Competition between the weed and the crop. You cannot separate these. But these are some of the technical issues. Here we've got a student who was doing weed competition in maize and Tithonia rotundifolia. This guy is currently in Korea, I think South Korea. Here we've got Tithonia rotundifolia with maize. You can hardly see the maize in that experiment. Here Tithonia rotundifolia under field conditions. Coming. Yeah. Excuse me, I just want to sign something. Okay, thanks. We want to talk about the weed control thresholds in Southern Africa. There's very little research which has been done on weed control thresholds in Southern Africa. The first flush of weeds in summer appears to exceed weed control thresholds. Therefore, weed control is always necessary. The second and subsequent flushes of weeds in summer appear to be below weed control thresholds. Then weed control thresholds in Zimbabwe. In winter cereals, in most cases, weed seedling emergence appears to be below the weed control thresholds. I think I may have to stop here.